Hello everyone. Today I will present my recent publication of myeloid trisomy 21 associated gene variant is protective from Alzheimer's disease. So first, I will give a background introduction about this publication. So, as you may know or not, so our lab is studies Down syndrome disease. So Down syndrome is caused by trisomy of human chromosome 21. People with Down syndrome tend to age more quickly. And one of the biggest challenges they face is the Alzheimer's disease. This happens because the extra chromosome in Down syndrome cause the body to make too much of the protein called amyloid precursor protein. When that builds up, it leads to beta amyloid plaques in the brain. So by their 40s, almost everyone with Down syndrome show Alzheimer-like changes in their brain. And by their 60s, about two-thirds are diagnosed with this disease. And even though most DS individuals show these brain changes, a small group that don't go on to develop dementia. So somehow they remain cognitively healthy because despite having the same pathological signs. So this may raise a fundamental question. So why does the neurodegeneration occur in some brains but not others, even under the same pathological burden? So um, in Alzheimer's disease, there's also a subset that don't develop, they have the pathological proteins, but they also don't develop the dementia. So to study the what protects them, and it could help us design new treatments. So not just for Alzheimer's disease, as well as for the DS community. Um, Traditionally, so Alzheimer has been viewed as uh, as well as a neuron-centric disease, but in recent years, evidence has shifted our attention toward to the immune system. So there are two studies showing the opposite result. The myeloid cell, when they penetrate the brain, they can cause the neurodegeneration disease. And there's another study which indicated that the clonal hematopoiesis, for example, is associated with reduced Alzheimer disease. These myeloid mutations arise in the blood, can enter the brain, which is influence the neurodegeneration. And these observations lead us a provocative idea. So what if resilience to Alzheimer's disease originates at least in part from the hematopoietic derived cells, especially microglia? Then we want to ask why microglia. So microglia uniquely present to shape disease outcomes. They are long-lived, self-renew, and they are very sensitive to pathological proteins. They can also promote synapse loss or protect synapse, and they can propagate the inflammation and or suppress it. So instead of asking why microglia contribute to Alzheimer's disease, we may want to ask a different question. So can microglia actively protect the brain from the Alzheimer pathology? So next I want to introduce some information about this hot mutation, CSF12B. So there's a paper from 2019, which introduced a gain of function mutation, CSF12B. So what is CSF12B? CSF12B encodes a shared cytokine receptor subunit for IL-3, IL-5, and GMC CSF. These are key regulators of myeloid survival and also activation. And these signals can go through the step 5, PR3, PI3K, AKT, and ERK. molecular pathways. And this gain-of-function mutation has been identified in the DS individuals, and this mutation can drive the ligand independent STAT5 activation, and is and also but its role but the role of this mutation in human microglia, particularly in the Alzheimer disease, was completely unknown. Oh. And there are also some studies that introduce the GM-CSF, their protective effects both in the Alzheimer disease and as well as the Down syndrome disease. Therefore, we have our hypothesis: the microglia carrying the trisomy 21 associated gain-of-function mutation, which may impact the resilience in Alzheimer disease. So next, I want to introduce the result of my study. So first, we introduce this mutation because this mutation I identified from the Down syndrome individuals. Therefore, our first question we want to ask: if we introduce this mutation in the Down syndrome, if we introduce this mutation in the Down syndrome, if they have the protective effects. So first, we introduce this mutation in the DS RPSCs. Then we differentiate this DS RPSCs into the primitive macrophage progenitors. Then we further promote the microglia maturation with these PMPs. So first, we use the in vitro experiments to explore the protective effects of this mutation. So when we have the microglia in vitro, so we challenge these cells with pathological proteins. So then after 24 hours, we evaluate their cell viability. We can see that in the when these DS microglia under the DS-editor conditions, the mutant the DS microglia carry this mutation, which they can see the resistant to this tau induced toxicity. When we block this STAT5, the AC4 113 is a STAT5 inhibitor. So when we block this signal, we can see this protective effect abolished by this inhibitor. So we can get the conclusion from the in vitro experiment that this mutation protects the iPSC-derived microglia against the pathological tau-induced the cytotoxicity effects. So next, I want to mention my previous study, so which published in 2022. So in this study, I identified the DS microglia can show the dystrophic and the senescent phenotypes when they respond to the pathological tau. And in this scenario, the type 1 interferon play a very critical role. So based on this study, so we further want to ask if we introduce this mutation to the DS microglia, if they can against the senescence. So first, we transplant this mutant microglia into the Neanderthal pups. And then after 2 months, we challenge this chimera brain with pathological tau. So first, we want to ask what kind of molecular mechanism underlying this scenario. So first, we collect the we purify the human microglia from this chimera mice, then perform the single cell RNA sequencing. So we identify the 8 cluster from this human microglia. So first, we perform the GSA senescence gene enrichment analysis, we identified the senescence-related genes are downregulated in the DS mutant DS-editor group. And when we perform the senescence score analysis, we found also the senescence-related genes are downregulated in the DS mutant DS-editor group. As I mentioned before, so the type 1 interferon play a very critical role in the DS microglia when the challenge with the DS editor. Here we notice the majority of the type 1 related genes are downregulated in this mutant microglia when the challenge with the DS editor group, which suggests us this mutation could protect the DS microglia against the senescence. To further verify the findings from the single cell, so we also did the microglia morphology analysis. So which we can see when the microglia carry this mutation, they have the improved morphology when compared to the Y-type microglia without this mutation. Also, they have the very lower ferritin expression, which is a senescence marker. And this finding also verified with P21 staining. P21 is the cell arrest marker. It can also indicate the senescence. So we notice when the microglia carry this mutation, they have a lower P21 expression. To further verify the type 1 related genes expression, we also use this human microglia to perform the qPCR. We find the type 1 related genes, for example, IRF8 and IFI-TM3 are downregulated in the mutant microglia group. So secondly, we also find this mutation, the microglia carrying this mutation, they also could develop a protective microglia subpopulation. So in this among these 8 subclass of this human microglia, we identified the class 3 and the class 6 are all DEM class, the disease-associated class. However, class 6 only exists in the DS carries mutation group. So when we next perform the trajectory analysis, we find the starting point are from the homeostatic class 0, and the end point is the 2 DEM class. The lineage 1 is correspond to the class 6, and the lineage 3 is correspond to the class 3. So then we perform the go-term analysis corresponding to the different development stage. We notice the class 6, which are only existed in the DS microglia carries mutations that highly respond to the protective term. For example, the plasma memory repair and the tissue remodeling, and also the wound healing, which indicated the protective effects. Even there is the disease associated class, but they have the protective effect. Then we want to ask, because the previously single cell time point is we collected after the injection for 2 months. So next we want to ask if this protective disease-associated microglia identity can prolong after several across different time points. Then we also collected the chimera brain about 8 months after the TAU injection. So we also identified the 8 microglia cluster. And among these clusters, there are 4 homeostatic clusters. And the cluster 3 is corresponding to the previously protective cluster 6. And also the disease-associated cluster, which are similar to previous cluster 3. And the cell cycling cluster both social cluster. And when we perform the cell proportion analysis, we notice the majority of the cells in the 10 months samples are homeostatic clusters, and the type 1 response gene cluster are nearly gone in the in the 10-month samples, which indicates that this protective disease associated microglia, they have the long-lasting role in after the tau challenge. Then next question is we want to ask if this microglia, they also have the neuronal function, protective neuronal functions against this tau injection. So we did 2 experiments. So first is the DCX staining. We noticed in the DS microglia carrying this mutation, they have a comparable DCX expression to the control group. And also we performed the LTP recording. We noticed the DS microglia challenge with the DS editor, they have the very similar FEP slope compared to the Y-type group. So because the previous experiment we introduced the mutation to the DS microglia and already proved their protective effects. So next we want to ask if these mutations are independent of trisomy 21. Then we performed we introduced this mutation into the 2 different control iPSCs. Then recapitulate the several key experiments. So first we did the microglia morphology analysis. We noticed the microglia morphology in the control DS control mutant microglia. They have the similar morphology compared to the control microglia. And also we did the senescence marker staining, and also as well as DCX staining, and also the functional analysis, the LTP recording. We noticed when the control microglia carry this mutation, then after carry this mutation, they have the also the resilience effects under the pathological tau conditions. So so next we want to ask if we put these 2 different microglia. So one is carrying this mutation, one is without this mutation. We put this microglia in the same environment, then challenges the tau protein if they have how about their reactivity in the same condition. So then we use the CAGG to label the normal microglia and introduce this mutation to the cell without this without the GFP fluorescence that do the co-transplantation. So before we do the co-transplantation, we use the 1-to-1 ratio to mix these 2 different kind of cells, then challenge this tau protein. So first we check the cell distribution. We found after 4 months tau injection, so both of the hippocampus, they have very good repopulation of these human cells. Then first we compare the different ratio of these cells. We notice in the DS-ID tau condition, so the control, the GFP level Y-type microglia, they have the slightly decreased proportion. However, in the microglia carry this mutation, they have increased proportion. And also we did IBA1 human nuclear double staining. We noticed the same result. And secondly, we also noticed in the this mutant microglia, when we perform the 3D reconstruction analysis, we notice this mutant microglia that can engulfing the Y-type microglia, which we see the GFP-labeled cell debris inside the mutant microglia. Then we collect the microglia to perform the single cell analysis. We notice in the DSID-TAL condition, the mutant microglia, which means the microglia carrying this mutation, they can actively engulf the GFP-labeled Y-type microglia. And there are many phagocytosis-related signal pathway. For example, the CD14 TLR4 and the PLAT LRP1, they only enriched in the mutant microglia, which indicate and this ligand receptor not enriched in the GFP labeled microglia, which indicated a unidirectional unidirectional phagocytosis signal pathways. So to summarize three different parts. So first the CSF2RB F150D mutation that provides the protection to microglia against the tau-induced toxicity in vitro. Then we use the camera also protect this, also proved this mutation that can confer the protection microglia against the senescence and also the protects the neuronal functions against this pathological tau. And also this resilience effects conferred by this mutation, they are independent of trisomy 21. And lastly, we proved that the control microglia which carry this mutation that can provide the protection against the tau conditions, so the phagocytosis, so the replacing this Y-type microglia. Yeah, that's all. So lastly, I will thank our lab members, also all our collaborators, the funding from NIH, also my fellowship funding. Thank you.